Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Chantelle Gaskin, Dr. Ilaria Scarfone, and Dr. Julia Baek from Thermo Fisher Scientific.
Thanks, Leah. The gene therapy landscape has exploded in the last several years, bringing a multitude of viral vectors to the clinic. At the forefront of the gene therapy clinical landscape is the application of lentivirus, adeno and adeno-associated virus, and plasmid-based therapies targeting all types of genetic diseases. With each program that progresses through the clinic, the body of manufacturing and clinical knowledge grows, and therefore so does the availability of regulatory guidance. It's this increase in regulatory guidance that drives the need for companies like ours, Thermo Fisher Scientific, to develop efficient and convenient purification and analytical tools for our customers. Here at Thermo Fisher Scientific, we value our ability to support your gene therapy program. From plasmid development through viral vector production, purification, and analytics, we have a vast portfolio of products and services, as well as a team of cross-functional bioprocess experts to provide solutions throughout your process. In this webinar, we'll focus on downstream purification and analytical methods relevant to the gene therapy workflow. Let's take a quick look at the gene therapy workflow, specifically for an AAV vector production. As with any bioproduction process, it is critical to monitor production quality and purity throughout the entire process using robust analytics. Starting with plasmid production through viral vector production in the upstream, regulatory guidance recommends monitoring non-vector DNA and mycoplasma, as well as other contaminants to aid in optimization of the process. In the downstream, purification process development scientists are increasingly focused on empty capsid removal and adventitious viral clearance, in addition to the standard host cell protein and DNA reduction requirements coming out of the regulatory agencies. And finally, release of an AAV product also includes meeting specifications for residual host cell DNA and plasmid, among others, which we'll touch on next. Here is a abridged list of typical quality and release assays for an AAV drug product. Most release assays can be categorized as either strength, safety, or purity assays. Some viral vector programs will have additional viral protein and DNA residual assays, among others, that are not called out here, but today we'll cover a range of quality assays that we support, including mycoplasma, host cell DNA and plasmid quantitation, total capsid titer determination, viral clearance studies, and empty full capsid analysis. Taking a look again at our AAV production process workflow, it's critical to perform mycoplasma testing in the upstream bulk harvest material prior to progressing through the downstream process. Ilaria will now walk us through the mycoplasma testing process.
Thank you, Chantelle, for the introduction. So, mycoplasma is a known contaminant in mammalian cell culture and can affect the safety, quality, and efficacy of biotherapeutic products. Because of this, it is a regulatory requirement to demonstrate that cell culture-based therapeutics are free from mycoplasma. Previously, mycoplasma tests were based on a 28-day cell culture method. We've seen over the last 10 years an industry-wide move towards alternative nucleic acid techniques, and today, rapid real-time PCR methods, such as our MycoSeq mycoplasma detection kit, are globally accepted by regulatory bodies for Mycoplasma lot release testing. In order to maximize the sensitivity of safety testing, regulatory guidance recommend that Mycoplasma testing is performed when contamination is most likely to be detected, and mentions PCR-based tests as examples of alternative methods for Mycoplasma testing after a suitable validation. MycoSeq Mycoplasma detection system has been specifically designed to meet regulatory lot release expectations. MycoSeq is a fully integrated real-time PCR-based system. The system features are MycoSeq Mycoplasma detection assay, QuantStudio 5 real-time PCR instrument with specifically developed AccuSeq software, and PrepSeq nucleic acid extraction technology with manual and automated extraction workflows. The MycoSeq detection assay has been designed to fulfill regulatory requirements, and following validation, regulatory filing, and review, many customers have received regulatory acceptance from different regulatory agencies to use the MycoSeq assay for lot release testing applications across multiple therapeutic modalities. This includes cell and gene therapy applications, monoclonal antibodies, recombinant proteins, and vaccine production. There are currently over 30 customers going through the process of MycoSeq validation and regulatory submission to use the assay as a lot release test for their drug products. The MycoSeq kit sits as part of a complete end-to-end solution that allows the same-day Mycoplasma contamination results. Here is an overview of the full workflow. It starts with nucleic acid extraction using our well-established magnetic beads-based PrepSeq chemistry. Extraction can be performed manually, or you can use one of two automation systems. The Automate Express, pictured here, for fully hands-free automation, or our semi-automated Kingfisher Flex instrument for high-throughput users, allowing up to 96 extraction per run. Next is the real-time PCR assay using our MycoSeq kit, which uses cybergreen detection dye and a multi-primer approach to detect over 140 different species of Mycoplasma and related species. Its sensitivity has been demonstrated by internal and external validations to detect fewer than 10 Mycoplasma genome copies. Our latest recommended instrument for detection is the QuantStudio 5 PCR system with AccuSeq software that allows automated presence-absence calls. The full workflow enables you to deliver results in less than 5 hours to allow the rapid and early detection of Mycoplasma. As the MycoSeq assay uses cybergreen detection, we can draw on 3 objective analysis parameters: cycle threshold to show the amount of a target DNA present in a sample, melting temperature and derivative value from the melt curve analysis to show the size and amount of target amplicon produced. These criteria, established by the customer during validation, must be met for a sample to be positive for the presence of Mycoplasma. One thing I'd like to highlight at this point is that because MycoSeq facilitates a multiplex PCR, it means that outside of controls, you only need to run one PCR reaction per sample. And because running only one multiplex PCR for Mycoplasma detection is such a separator, MycoSeq has received a patent for its multi-primer approach. This slide shows the partial inclusion panel for the MycoSeq Mycoplasma detection assay. The MycoSeq kit is capable of detecting over 140 Mycoplasma, Aceloplasma, and Spiroplasma species with no cross-reactivity to closely related bacteria. We have directly wet lab tested 46 different species, including the ones commonly used in validation studies, here shown in red, and the rest of the species have been determined bioinformatically. Actually, the MycoSeq assay has received a second patent for the discriminatory positive control that is included in the kit. The discriminatory positive control, or DPC, facilitates the amplification of a modified PCR product with a melting temperature well outside the range of real Mycoplasma amplicons. Using post-PCR melting analysis, you will always be able to tell whether a sample contains real Mycoplasma or your positive control. At the same time, the DPC maintains the same extraction behavior as genuine Mycoplasma DNA and can be used as a sample extraction positive control by spiking test samples without the risk of unrecognized cross-contamination. Furthermore, we use the DPC as an inhibition control at the PCR level to determine whether sample matrices have any impact on the PCR itself. Therefore, the DPC significantly reduces the possibility of both false positive and false negative results. So because of these factors, the DPC can be used as a risk-free surrogate for Mycoplasma DNA or live Mycoplasma material during method optimization and early qualification. Finally, using the Applied Biosystem AccuSeq real-time detection software, you can generate automated presence-absence results during data analysis. Automated calls are determined based on the cycle threshold, melting temperature, and derivative values of the test sample and the inhibition control. This software is integrated with our 7500 FAST instrument and with the QuantStudio 5 real-time PCR. AccuSeq also has security, audit, and e-signature capabilities that are designed to enable 21 CFR Part 11 compliance required in a GMP environment. To summarize, the MycoSeq Mycoplasma detection system is a highly sensitive and specific real-time PCR assay. Our unique discriminatory positive control is used to confirm extraction and PCR performance without the risk of false positive by accidental cross-contamination. MycoSeq has received regulatory acceptance as lot release test for a variety of on-market therapeutics across various modalities. And finally, the level of support we provide will enable customers to confidently progress through assay optimization, validation, and a regulatory submission. Okay, so with the confirmation that the bulk harvest is Mycoplasma free, the Clarify harvest is now ready for downstream processing, and Chantelle will go into the detail.
Thanks, Ilaria. The first purification step in the downstream AAV process is the capture step, commonly using affinity chromatography. So next, I'd like to introduce to you our affinity resins that make up our Capture Select technology brand. Capture Select ligands are camelid antibody fragments produced recombinantly in yeast, making them animal-derived product-free so they're compatible with a commercially viable process. Our AAV affinity ligands are immobilized onto our porous base bead, which has excellent characteristics for scale-up, and we have 3 AAV affinity ligands. 2 of them are serotype specific for AAV8 and AAV9, and the third, porous AAVX, is considered a universal capsid affinity resin. It's been developed to provide high capacity and specificity, and through internal studies and customer feedback, we have confirmed that AAVX is capable of greater than 90% recovery and over 90% purity from a single step. All of our Capture Select products are also available as free conjugated ligands. Our AVX ligand is available in biotinylated and HRP labeled formats, which our customers have used to develop in-house ELISAs to determine total capsid titer in both in-process and purified samples. For example, during process development, you may benefit from determining the total capsid titer in your clarified harvest to appropriately size your affinity column. You might also want to use AVX capsid ELISA to monitor the mass balance in your harvest and capture steps. Next, we'll show us an example of how our AVX conjugated ligands can provide a simple approach to developing a highly sensitive capsid titer assay, as seen in this cartoon here at the bottom, using streptavidin-coated plates to bind the biotinylated capture ligand and HRP-labeled detection ligand. In this example, we detail a total capsid ELISA for multiple AAV serotypes, where we use AVX conjugated ligands to develop a 4-step assay. The biotinylated AVX ligand is diluted and aliquoted onto streptavidin-coated wells and then incubated. Standard curves of each serotype were prepared separately for AV1, 2, 5, 6, and 9, and then the standard curves were then aliquoted onto the coated wells. After a 1-hour incubation, the wells were treated with a diluted preparation of the anti-AVX HRP detection ligand, and this is followed by a 10-minute incubation with TMB substrate, and then the reaction is stopped with acid. This method is intended as a starting point, and of course, in-house method optimization is always recommended. But under most conditions, you can expect a valid assay in the ranges of 1E8 and 1E11 capsids per mil for most serotypes. As I mentioned earlier, another guidance coming from regulatory agencies is the recommendation to determine the viral clearance capability in each unit operation of your AAV process. In addition to host cell and process-related impurity clearance, our AVX resin has been shown to provide effective viral clearance. In this example of a viral clearance study from REGENXBIO, where their AAV8 process includes an AVX column as the capture step, this process is tested using a 6-virus panel of enveloped and non-enveloped viruses, as well as RNA and DNA containing and viruses of all of different sizes. What we concluded from the study is that AVX can achieve greater than 4 log reduction of 4 out of the 6 viruses tested, and between 1 and 3 log reduction of the remaining 2 viruses on the panel. Other biopharma companies have published their viral clearance data, further suggesting that AVX can indeed be a powerful tool for viral clearance in your downstream process. Another important analytical tool to evaluate AAV product purity at this point in the AAV workflow is empty full capsid analysis. One of the methods used is HPLC analysis. Julia will now take us through her method development for empty full capsid analysis by HPLC.
Thanks, Chantelle. So HPLC analysis of full and empty particles can be done after affinity purification and after further polishing purification to check the effectiveness of your anion exchange removal of empty particles. And HPLC method requires less sample than analytical ultracentrifugation, and also you can get your data in 20 minutes or less, depending on your sample and chromatography method. I have tried several different HPLC modes and found that anion exchange chromatography works well for AAV full and empty particle separation. And you can find several publications using anion exchange chromatography. At Thermo Scientific, we a ProPak SAX10 column, which is packed with a polymer resin that is coated with a hydrophilic layer to prevent unwanted hydrophobic interaction, and also has grafted polymer chains that carry anion exchange functional groups. Our SAX10 column has strong anion exchange groups, which is a quaternary amine group. When you run anion exchange chromatography for protein and AAV particle analysis, you can use salt gradient or you can use pH gradient. With salt gradient, the protein sample binds to the stationary phase through charge interaction, and as you increase the salt concentration, the protein elutes. With the pH gradient method, the protein is negatively charged at high pH, and in this case, AAV particles are negatively charged. And as you decrease the pH, the AAV particles become neutral at its PI value and elutes from the column. There is a small difference between the PI of full and empty particles, which allows this separation. Here I will talk a little bit about method development and chromatography settings when you run AAV particles. First of all, you have two choices when it comes to detection. With UV, you can get full and empty information by looking at the intensity of the signal at 260 and 280 nanometer wavelengths. For full, you would see higher 260 nanometer signal, and for empty, you would see higher 280 nanometer signal, as you can see in the chromatogram shown here on the right. However, the sensitivity of UV is lower compared to fluorescence, and also fluorescence provides better quantitative data since you are only monitoring the AAV capsid signal, which comes from the tryptophan residue of the capsid proteins. For mobile phases, I have tried multiple different conditions and found that bis-tris propane as buffer and tetraalkyl ammonium chloride as salt works great, as also shown in this paper, the first paper listed here in the slide. The optimal pH will depend on the serotype, at least that's what I have seen. So I recommend trying both pH 8.5 and 9.5. For tetraalkyl ammonium chloride, tetraethyl ammonium chloride gave better resolution than tetramethyl ammonium chloride, but I found that it has higher background signal. This is probably due to impurities in the salt. So along that point, for all of the chemicals in the mobile phase, I recommend you to make sure that you purchase high purity grade so that you can get a clean background. For pH gradient, here are mobile phases we found to separate full and empty particles well. Mobile phase A, pH is 9.2, and mobile phase B, pH is 2.8. So if you run 100% A to 100% B, you would get a pH gradient from 9.2 to 2.8. Once you run that and figure out where your sample elutes, you can shallow the gradient to get better separation. But I would also like to note here that for some samples, this pH gradient method did not elute the AAV particles. This is not necessarily serotype dependent, since I have seen same serotype, different prep, giving different results. So at this moment, we are not sure why. One possibility we can think of is there are some host cell DNA stuck on the surface of the capsid. And since DNA charge would not change during the pH gradient, the sample may be stuck on the column. So for those samples, we recommend using salt gradient. For temperature setting, I typically set it at 30 degrees, but you can also explore higher temperatures. But keep in mind the stability of the capsid structure at the temperature and the pressure you are analyzing. With these chromatography conditions, we were able to separate multiple different serotypes, and here we have two examples using salt gradient, AAV6 and AAV8. In both cases, we got baseline separation of full and empty. For these examples, I used tetraethyl ammonium chloride as salt. And you can see for the AAV8, we were able to separate a little bit more than just empty and full. I do not know exactly what that middle peak is. Maybe it's partial, maybe it's broken particles. We don't know, but we were happy to see that we separated something more than even empty and full. And with a pH gradient, we were also able to get good baseline separation, as you can see with these AAV8 and AAV9 chromatograms. And also note here that for AAV9, the empty sample elutes later and the full sample eluted earlier. And I've seen this with at least 2 different preps of AAV9. So that could be a general rule for AAV9, but more study needs to be done. Here, this slide shows excellent lot-to-lot reproducibility of our ProPak SAX10 column. Full and empty ratios were consistent between these 3 different lots that we have tested. So in summary, using the ProPak SAX10 column and optimized methods, you can get good separation of full and empty particles, which allows measurement of full and empty ratio of your sample. And this measurement confirms a successful upstream AAV production process and also provides a baseline for further downstream purification. Now Chantelle will talk about removal of empty particles using porous anion exchange resins.
Thanks, Julia. Next, I'll talk about the empty full capsid separation polish step. But first, a little bit about our porous resins. Our purification anion exchangers are manufactured using the 50 micron diameter porous base bead. The bead is made of polystyrene divinyl benzene, which provides rigidity for a stable column bed and enhanced stability, chemical stability. These resins provide high binding capacity and linear pressure flow curves without compromising on resolution as you scale up. In this sample chromatogram here in the middle, we took a mixture of 3 proteins and applied them to a porous column. As you can see, as we ramp up from 100 to 1,000 centimeters per hour, the resolution is maintained. This is what makes our porous anion exchangers ideal for developing an at-scale full capsid enrichment polishing step, because the difference in PI between empty and full particles is so negligible that high resolution really is necessary to separate the 2 species using a conductivity-based elution. We carry 4 anion exchangers in our portfolio with a range of weak and strong base chemistry. Porous D and PI are our weak anion exchangers. Porous HQ and XQ are the strong anion exchangers. They also come in a variety of pore sizes and structures, allowing for increased mass transfer and high capacity, which is ideal in a large biomolecule process such as AAV. Our most popular resin for empty full capsid separation in a full-scale process is porous HQ. Here we have a published example of porous HQ in an empty full capsid separation process. In this paper by Chu et al., we see that empty AAV2 capsids purified by cesium chloride gradient are applied to a porous HQ column and then eluted by a linear sodium acetate gradient. The same was done with full capsids purified by cesium chloride. We see that here on the top, empty capsids characterized by a higher absorbing A280 peak elute sooner than the full capsids characterized by a higher absorbing A260 peak on the bottom. When the empty and full capsids are combined at a ratio of 16:1 and applied to the same column with the same gradient conditions, the empty capsid peak presents first, followed by the full capsid peak with baseline separation, demonstrating the resolving power of the resin and also providing a feasible and scalable AAV empty capsid removal process. Another traditionally important function of downstream purification is the removal of residual host cell and vector DNA impurities. Next, we'll hear from Ilaria again, this time to talk about some simple and reliable analytical kits to measure residual DNA designed to meet regulatory requirements for lot release testing.
Thank you, Chantelle. Yeah, so in a classical adeno-associated virus production workflow, viral vector production depends on the availability of high-quality plasmids. However, these plasmids are not allowed in the final product and thus need to be removed. This creates the need of a sensitive and robust method to measure residual plasmid levels. In the downstream process, AAV are purified. Here is when a residual host cell DNA and plasmid DNA quantitation is required. Residual DNA quantitation may be performed at various stages throughout the downstream process, starting with the cell culture harvest all the way down to the final product, in order to monitor DNA clearance. So the reason why you should be concerned with the amount of residual DNA left in your product is that we know that DNA can impact product quality, efficacy, and safety. Regulators around the globe therefore require you to limit the amount of residual DNA in the final dose. The World Health Organization, for instance, requests that the amount of residual DNA per dose is kept below 10 nanograms. Residual plasmid is part of the 10 nanogram per dose limit along with host cell residual DNA. The FDA requests that the host cell DNA content is kept as low as possible and that you are using a highly sensitive method to determine DNA level. The FDA also suggests that you conduct rigorous clearance studies throughout the downstream process to show that the process is capable of removing the vast majority of DNA from the product and to monitor process deviations. Currently, we offer a variety of host cell lines for residual DNA testing, including E. coli and the kit specific for SF9 and baculovirus AAV production system. Our newest addition to the portfolio is the kanamycin resistant gene assay for the quantitation of plasmid DNA. I will focus my presentation on this particular kit and on the HEC293 kit, as HEC293 cells are typically used for AAV packaging. However, most of what I say about this kit is also true for all the other res-DNA-seq flavors. The Applied Biosystem res-DNA-seq assay is a fully integrated real-time qPCR-based system for the quantitation of residual DNA. The system includes highly characterized DNA reference standards. The key features of the assay are ultra-high sensitivity with a limit of quantitation of down to 0.3 picogram per reaction for HEC293 DNA and 30 copies for kanamycin plasmid-resistant DNA, a rapid and streamlined workflow with optional sample preparation automation and time to results of less than 5 hours, and a reliable performance that provides consistent results from kit to kit, lot to lot, and year to year. The streamlined workflow begins with sample preparation, where you can choose between manual and automated options. The option shown here is a semi-automated version, utilizing the Kingfisher Flex for up to 96 extractions at a time, meaning that you could easily go through 500 samples per week. Regardless of the level of automation chosen, the sample preparation always features the well-established PrepSec chemistry, which is based on magnetic particle separation. The second and third step in the workflow is setting up and running the res-DNA-seq assay on one of our recommended Applied Biosystem real-time qPCR instruments. The AccuSeq real-time PCR detection software integrates with our recommended instruments and provides many features to support you with the setup, running, and analysis of your real-time PCR experiments. The software has assay functionality that helps enable 21 CFR Part 11 compliance and full traceability of all action within the software. This slide gives a quick overview of some assay specifications. You can see the specification for the res-DNA-seq quantitative plasmid DNA kanamycin resistant gene and HEC293 residual DNA kit. In conclusion, these are a high assay linearity as given by the R-squared value of more than 0.99, a high PCR efficiency of around 100% with a maximum variation of plus minus 10%, an assay precision with a maximum of 10% CV, a limit of detection of 15 copies for kanamycin resistant gene kit and 30 femtograms for HEC293 DNA kit, a limit of quantitation of 30 copies for kanamycin resistant gene kit and 300 femtograms for HEC293 DNA kit, and the linear assay range from 30 to 300,000 copies per reaction for kanamycin resistant gene and from 300 femtograms to 3 nanograms for HEC293 residual DNA kit. The assays have been validated using both our recommended instruments, the 7500 FAST and the QuantStudio 5. In addition, all of our DNA-seq assays have been also shown to provide accurate results with various sample matrices. Here, the graph on the left shows the standard curve generated using the serial dilution of plasmid DNA standard provided in the kit. The dynamic range reaches from 30 to 300,000 copies. The second one shows the standard curve generated using serial dilution of HEC293 DNA standard provided in the kit, ranging from 300 femtograms to 3 nanograms. These data prove the high sensitivity and broad dynamic range of these two assays. The broad dynamic range allows testing a wide range of samples. You can see that the method linearity within the dynamic range easily meets the specification as demonstrated by the R-square value above 0.99. In addition, the PCR efficiency has been calculated to 105.3% and 101.6%, which sits well within the range of 90 to 110%. On this slide, I would like to take a look at recoveries. The kanamycin-resistant gene plasmid DNA assay has been designed to detect and quantitate the vast majority of kanamycin-resistant gene alleles currently known. This has been achieved by carefully analyzing the conserved region and developing a multi-primer assay to target all alleles with the same sensitivity. This table is showing 3 different commercially relevant plasmids with the respective recoveries from a typical sample matrix. The plasmids were spiked into the matrix at 100 and 10,000 copies, manually extracted using the PrEP-seq chemistry, and quantified using the kanamycin-resistant gene plasmid DNA kit. As you can see, all plasmids were detected with a recovery greater than 85%. Very similar results in terms of DNA recovery rate can be observed from the validation data of the HEC293 kit. And here is the data showing the specificity of the assay. To demonstrate the specificity of the kanamycin kit, we have performed a series of experiments with unrelated DNA directly spiked into the PCR reaction. On the left, you can see a table showing the type and amount of DNA spiked into the PCR, including most of the relevant source in bioproduction. The graph in the middle shows the internal PCR control included in the assay to monitor PCR inhibition. All reactions provided the same CT value, indicated the all PCRs performed as intended. On the right, you can see the amplification in the target channel as given by the recorded CT values. The first lane in this graph shows the amplification of the DNA standard included in the kit at 15 copies, providing a CT of around 34. Most of the other reactions provided no amplification curve at all. Others provided a range of CTs well above the limit of detection. Together, these data should give you a first impression of the performance of the ResDNA-seq system. And with this, I would like to summarize our residual DNA quantitation solution in AAV production by highlighting once again the key features of the system. The ResDNA-seq system is an all-inclusive system coming with highly characterized DNA standards and all reagents necessary for residual DNA quantitation. It provides an optimized sample preparation for quantitative DNA recovery. It provides ultra-high sensitivity and specificity with no cross-reactivity to unrelated species DNA. The streamlined workflow provides reliable data within 5 hours. Our quality assurance team is making sure you will be receiving the same high quality performance from kit to kit, lot to lot, and year to year. And finally, our worldwide technical support network will support you throughout all phases of the implementation process, from early evaluation over qualification and validation all the way to routine testing. In conclusion, we hope this was an educational presentation of only a few of the purification and analytical tools Thermo Fisher has developed to address and simplify downstream purification and analytical AAV manufacturing challenges. As current strategies, techniques, and regulatory guidance continue to evolve in the gene therapy space, Thermo Fisher is uniquely positioned to support with a large global footprint of research and development, production, and field support scientists, able to respond to the changing landscape and industry needs through consistent high-quality purification and analytical tools, including custom purification resins and analytical assays designed and verified to meet regulatory guidance in a cGMP environment. Thank you very much for your attention, and we'd be happy to take any question at this time.
Okay, great. So the first question is, in your AVX viral clearance case study, what can be done to improve clearance of an adventitious virus such as HSV and Rio3?
Thanks, Leah. I think that's for the purification side. You can begin using multiple wash conditions. So in this viral clearance study that we demonstrated earlier, it wasn't outlined what types of wash studies were included, but typically optimizing your wash studies is the first route. But there's sometimes where you won't be able to add more than 1 to 3 logs of viral clearance just in the AAV step. So in those cases, you will have to rely on your other unit operations to clear the bulk of the viruses in the case of, for example, Rio3 and HSV.
Okay.
You might also, I'm sorry, just real quick, you might also want to add a virus filter step after your second column in addition to that.
Okay. And how can you tell if there's matrix inhibition when using the MicroSeq assay?
Yes, that's it for me. Thank you for the question. So for MicroSeq, we normally ask to run a matrix inhibition control along with your sample. So this is basically your extracted DNA with the addition of 2,000 copies of DPC, discriminatory positive control. So you can compare the CT that comes from the inhibition control to the one that comes from the positive PCR control, which contains only master mix and DPC. And you can detect if there is any inhibition that is given from your matrix.
Okay. And how much sample do you need for HPLC analysis of full and empty particles?
Sure, I'll take that question. It will depend on the sensitivity of your fluorescence detector and your detector setting. I typically inject 1 microliter of a 1 × 10 to the 13 Vg per ml concentration sample, and I have my detector set at the highest sensitivity setting, and also the temperature, I set it at room temperature. So you have to note that the higher the temperature, your signal sensitivity is going to go down.
Okay. And is there any time difference between manual and automated sample prep in the MicroSeq or ResDNA-seq workflow?
Well, the difference is not so much in the time needed to complete the workflow, but in the amount of analyst hands-on time required and the number of samples that can be processed at one go. So in general, all sample preparation workflows require roughly 2 hours to complete. However, while the hands-on time for the manual workflow is pretty much the entire 2 hours, the hands-on time for the Automate Express workflow is in the range of 10 minutes. The Kingfisher Flex workflow sits in the middle with about 30 minutes hands-on time. And In relation to the throughput, the manual workflow can handle up to 16 sample extractions at the time. For the fully automated Automate Express, the maximum sample number is 13, and for the Kingfisher Flex workflow, the maximum sample number is 96 per run.
Okay. In your example of AAV polishing step, a linear gradient was used. Can a step gradient be performed for separation of empty and full capsids using the porous AEX resins?
Yes, thanks for the question. Yes, it is possible to perform a step gradient using our AEX resins. So it's important to note that the empty and full capsids have very close PIs, and so the elution conductivity is usually only separated by, you know, a handful, maybe 1 to 5 millisiemens or so. And so that obviously is going to make it more difficult to develop a step gradient. But there have been a couple of publications that were released this year, and they're focused on working around the problem by optimizing buffer components so that they can modulate the elution conductivity between the two capsid species. And in one of those papers, the authors used a divalent buffer salt as well as sodium sulfate to achieve baseline separation between empty and full capsids in an AAV5 process. And that was using a porous HQ.
Okay. And can I use the MicroSeq or ResDNA-seq assays on other PCR instruments? And would they work on a Quant Studio 7, for instance?
Yes, so you can use, in principle, you can use any applied biosystem qPCR system that provides the correct set of optical filters and the adequate block size for the PCR reaction. However, you have to know that AccuSeq software is only available for 7500 Fast and Quant Studio 5 instrument. And only when using the AccuSeq software you will be able to utilize the full potential of automated calculation and presence-absence call, and also the SAE functionality that enable 21 CFR Part 11 compliance.
Okay. And what column format do you recommend for HPLC analysis of full and empty particles?
I recommend using the 50 millimeter length column. You can try the longer 250 millimeter format that we offer, but your analysis time will increase, and I have not seen a lot of improvement with the resolution, so I recommend 50. But if you have a challenging sample and time is not an issue, you can try the 250 millimeter length column.
Okay. What types of sample matrices were used in the validation study of the HEK 293 and kanamycin resistance and ResDNA-seq assays?
So we have used pseudo matrices from the harvest, purification, and a final bulk stage of a typical gene therapy process, including additives like benzonase, excess of unrelated DNA, and detergents, and also typical cell culture media.
Okay, great. So as to the audience, just a reminder, if we don't get to your questions, we will be passing them on to our speakers. So please continue to type your questions in, and we will pass them along. The last question for the webcast is in your experience in the field, have you seen any matrix interference with the ResDNA seq kits? And if so, how were you able to overcome this challenge?
Uh, so I did not come across matrix interference with this particular kit yet. However, it is known that some additives, like for example high amount of heparin, are co-extract with DNA and can cause PCR inhibition. So I have seen this twice, and we were able to overcome the inhibition by intensifying the washing step during DNA extraction.
Okay. Well, thank you all. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing us a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.